In this lecture, I'm going to talk about the environment and how it affects brain aging and perhaps vulnerability to age-related neurodegenerative disorders. Also, as part of this is how the environment affects brain performance throughout life. And one way to look at this is from an evolutionary perspective. Animals typically, or perhaps almost always, evolve in environments where resources are scarce and there's competition for those limited resources. And one of the main resources is food. And so during evolution, individuals whose brains functioned well in a food deprived state had a survival advantage. And one can even speculate that, for example, certain higher order behaviors and capabilities of the brains of organisms evolved for the purpose of competing for successfully for limited resources and um, so that they could survive and reproduce. On the upper left, there is some wolves surrounding a buffalo, and and probably sometime during evolution, uh, individuals that cooperated more with their uh, mates or their pack colleagues, if you will, uh, were successful in being able to capture and kill large prey. So wolves can, by cooperating, can kill an animal that individual wouldn't be able to themselves. And then in human evolution, and I'm just kind of jumping here quick in evolution, just kind of some examples, kind of a thought type uh, type way to look at this. So as far as we know, all the early tools invented by humans were tools that had to do with acquiring and processing food. So, you know, just simple things like using stones to pound grain or break open hard nuts, uh, weapons for killing prey. And so one can kind of imagine that the capabilities of the brain that allowed humans to become inventive and creative in adapting to their environment and being able to acquire food and other resources gave them a survival advantage. And cooperation was part of that too. Okay, so fast forward to the modern area when many humans are overfed in terms of calorie intake. And as you know, obesity's become an epidemic in this country, including childhood obesity. And often people who have pets may overfeed them too. Uh, actually, it's kind of hard to overfeed a chicken, but they keep producing, if they're producing eggs, particularly. Um, right. So there's a lot of evidence. I'll show you just a few slides that this kind of poor metabolic health due to overconsumption of high caloric foods, relative lack of exercise, and perhaps somewhat the diet composition, addictive foods, high fructose corn syrup, uh, fats, and mixes of ingredients that are highly palatable. And these ultra processed food industries have chemists and others who develop these mixtures so that they're kind of addictive. Okay, yeah, so this is a big problem, and there's a lot of factors contributing to this. Uh, Big agriculture, monoculture, growing corn, soybeans, and wheat, for example, making the high fructose corn syrup from the corn. So a lot of these grains that are produced throughout the country are used to make ultra-processed food. And then food industry, of course, the pharmaceutical industry, there's no emphasis on risk reduction or prevention. They make money from people getting sick and then developing a drug to treat them. We need drugs to treat diseases, but there's no big incentive for a lot of industries to do that. And then this somewhat effort-sparing technologies. Many of us don't have to exercise at all to do our jobs, which is a big change in the last, well, particularly since the computer age, but even before then, some of the technologies that um, get us to the front door of where we work, and there's elevators and so on and so on. Okay, so this whole system is not good for the most part, and um, changes are needed. We all know that, including in medical education. I used to, before I retired, give a lecture each year to the medical students on essentially how good lifestyle habits are beneficial for the brain. And but that said, there isn't much on risk reduction and prevention in the medical school curricula. All right. So throughout the United States, there's a lot of variability in many health indicators. This map here, you could kind of superimpose it on a map for prevalence of obesity, diabetes, cardiovascular disease. That is to say, those common chronic disorders are more prevalent in these southern states. Okay, and then the reason I put this slide in here, and there's many reasons for this, why educational attainment is lower in these same states and, socioeconomic conditions, but also some of that has to do maybe with the fact that the metabolic state of these kids, more kids have obesity in these states, for example, and this is not good for the brain. And there's a number of reasons for that, including essentially promoting a lot of the age-related hallmarks of brain aging that I talked about previously. Reduced autophagy, uh, mitochondrial dysfunction, accumulated oxidative damage. Uh, One of the old theories of aging was simply that the more calories that come in to cells, the more free radicals that are produced in the mitochondria, and then that's causing the damage. But actually, that's probably not what's happening. It's that these poor diet and lifestyle factors are not activating adaptive stress response systems that enhance autophagy or mitochondrial function or upregulate antioxidant enzymes uh, or inhibit the mTOR pathway. Uh, if you're eating 3 meals a day plus snacks and have excessive calorie intake, particularly, then the mTOR pathway is likely turned on more chronically, which may be part of the reason there's impaired autophagy. Okay, but anyway, here's just from one study a long time ago, and there's many, many more studies like this in children, adolescents, and adults looking at their performance in various cognitive tests. This is a trail making test where they have to um, kind of a draw line by numbers thing. And see how fast they can do that. And here on the x-axis is the number of metabolic syndrome criteria. So metabolic syndrome is a combination of insulin resistance, abdominal obesity, hypertension, and of course, those are risk factors for cardiovascular disease and other disorders, including age-related cognitive decline and dementia. And this is another test that's kind of ability to concentrate. And then in this study, they did some um, MRI looking at size of the hippocampus and overall cerebrospinal fluid volume, so ventricular volume. Usually there's this relationship. I pointed out during aging, gray matter declines during normal aging, and ventricular size increases. So the skull has a limited volume. If gray matter decreases, then the ventricles typically expand. And so anyway, if you look at insulin sensitivity, there's a correlation between insulin sensitivity, that is good glucose regulation, ability to control glucose, and hippocampal size. And then there's a negative correlation with the cerebrospinal fluid volume. Okay, this is a slide, an interesting article where they're showing providing indirect evidence that a lot of that increase in human brain size among the primates uh, occurred coincidentally with the development, for example, of uh, inventing tools, uh, fire cooking, and those kinds of things. Okay, so that's all the bad news. The good news is that uh, age-related changes in the brain can be counteracted. All right, so here's a study from quite a while ago. Again, and many more studies like this. In this case, they're looking structural MRI. They had a control group that just did like light stretching, but not aerobic exercise, and then an intervention group. And they went over 1 year. And what you see, and these are adults, human adults, uh, size of the hippocampus increases with aerobic exercise and uh, no change or even a decline in the control group over a year. And then, and that's the brain region, at least among the ones they looked at, where they saw the biggest effect of the exercise. There wasn't a significant effect in thalamus, in the thalamus or caudate. All right, and it talked about BDNF and its role in neuroplasticity, synapse formation, mitochondrial biogenesis, and so on. And in this study, they show positive correlation between VO2 max, which is an indicator of aerobic fitness, a good one, and BDNF levels in the cerebrospinal fluid. So that's interesting. And animal studies have clearly shown that BDNF levels go up in many brain regions in response to, for example, running wheel exercise in rats or mice. That's enough for that. Now, this slide, so I have, I think, 3 slides that just list uh, an, many, many studies. And this is, oh, I think I forgot to put the reference in. This is from Henriette von Prague uh, and one of her postdocs. review article. I think it was in Cold Spring Harbor Proceedings or something like that. Anyway, it's a nice review article where they list some of the different studies in humans and animals that have looked at the effects of exercise in models or individuals with various disorders or humans with various disorders. And okay, so essentially, take-home message is regular aerobic exercise is good for the brain and may have some beneficial effects. Certainly, it's very good for improving mood, reducing anxiety, has an antidepressant effect. There's some evidence that BDNF plays a role in that by promoting neuroplasticity. Um, as far as is known, all of the treatments that are used to treat clinical depression, everything from electroconvulsive shock therapy, which isn't used much anymore, but still somewhat, to the serotonin, norepinephrine reuptake inhibitors, the most commonly prescribed antidepressants, they all increase BDNF levels in the brain. And some animal studies where BDNF levels are manipulated genetically suggest there's a key role for the upregulation of BDNF in the antidepressant effect. More recently, I should say also that this may be true for psychedelics, which are showing pretty good effects in initial trials in people with depression. So they may become, and the value with those over the most commonly used drugs is that there's long-lasting effect of one dose of the psychedelics like psilocybin, for example. Okay, so I'm not going to go in detail here, but you should know there's a huge amount of literature on exercise in various models of neurological disorders and in some human clinical trials. Okay, so this is just kind of another way to look at a slide that I presented before, where I had essentially aging here, and you could also put uh, poor metabolic health lifestyles like lack of exercise or overeating, not keeping your brain intellectually challenged, which is uh, good for your brain counteracting aging. So I'm going to talk about energy restriction, caloric restriction, because it's a really powerful way to extend lifespan in rats, mice, and other organisms and improve brain function and resilience. Uh, Exercise, intellectual challenges, and then I won't talk much at all about this, but there's some evidence that some of the chemicals in fruits and vegetables that are good for health, they actually, the reason they're in the fruits and vegetables is to act as, to dissuade insects and other organisms from eating those uh, plants. So these are kind of natural pesticides that we've evolved with, and they induce adaptive stress responses that may, for example, upregulate antioxidant enzymes. Okay, so these good lifestyle and diet factors uh, counteract biomarkers of brain aging, and a lot of this probably has to do with by enhancing adaptive stress responses. In fact, by imposing stress on cells. So I'm going to focus on neurons. There's been less looked at with glial cells. But the point is, these impose a stress, a good stress, a kind of stress that we evolved to not only tolerate, but in a sense, take advantage of because individuals who didn't evolve to take advantage of these stresses um, we're at a survival disadvantage. Okay, so I'll tell you that the, these good stresses, if you will, upregulate neurotrophic factors, DNA repair, enzymes stimulate mitochondrial biogenesis, protein chaperones, etc. Um, okay. Now, one thing I'll also mention, I'll mention it later, a recovery period is important. So obviously, you can't restrict energy intake too long because you get to a point of starvation. It's possible to overexercise. Um, we need sleep, and you can overdose on certain plants, although the ones we commonly consume, it's pretty hard to overdose on. So there are some potentially toxic chemicals in plants. Caffeine's one. Caffeine can actually kill you if you ingest too much of it. That's way below the amounts we take in are way below that. Okay, so these different things, lifestyle habits, there's a couple ways they may affect neuroplasticity and counteract brain aging. One is by affecting activity in neural networks. Um, for example, intellectual enrichment clearly is activating pathways, for example, into the hippocampus and then out to other brain regions and so on. Physical exercise, certainly in the sensory motor systems, there's increased activity, but also uh, apparently in other brain regions, including the hippocampus. Um, Also, there's humoral signals. So this has been looked at a lot with exercise. There's certain uh, proteins released, for example, from muscle cells or liver cells or perhaps other cells into the blood in response to exercise, and that then go to the brain and promote neuroplasticity. So I'll talk briefly about them. And the Canvas, just this morning in Canvas, I put up these lecture slides and I put up two articles. One was on exerkines, and then there was an article I wrote with some people in my lab a while back on pretty much what this lecture is about. These how transient exposure to good types of stresses or challenges uh, can promote neuroplasticity and counteract aging and neurodegenerative disorders. Okay, so exercise does a lot of things in the brain. The hippocampus has been one of the main brain regions looked at. It often is because of its prominence in learning and memory. It's uh, it's good organization of the neural networks in a way that, for example, you can make a brain slice and the entire circuitry is intact, so you can stimulate, say, dentate granule neurons and activate CA3 neurons or stimulate axons from CA3 neurons and activate CA1 neurons and so on. And also, it's a brain region where there's neurogenesis, one of two brain regions, the other is a subventricular zone. And exercise, and most of it's been running wheel exercise, voluntary exercise in rats and mice will stimulate neurogenesis. That was shown by Henriette von Prague in Rusty Gage's lab a long time ago. There's been a lot of work since. BDNF seems to play an important role in that. Um, and then in addition to BDNF going up, FGF2, fibroblast growth factor 2, which is a turns out very potent neurotrophic factor promoting the outgrowth of dendrites and axons and also promoting cell survival and protecting cells against various types of stress, excitotoxic, metabolic stress, um, and so on. Also, it's been shown that it's kind of similar to when you exercise your muscle cells, that it stimulates the growth of blood vessels into the muscle to provide, which makes sense, uh, which, over a period of time, say several weeks, a month of regular exercise, you get more blood flow because there's proliferation of the blood vessels, angiogenesis. And then exercise enhances learning and memory, particularly long-term running wheel exercise throughout life will attenuate age-related cognitive decline. So those are some of the changes going in the brain. Um, Now the question of whether any of these factors in the periphery are important for these changes or not. There's increasing evidence that they are. So here's some of the peripheral factors, and one that's not listed on here. Oh, this is the review article I talked about, this Cooper et al. 2018. Right. Okay, so from muscle cells, irisin is released, interleukin 6, a cytokine, and cathepsin B, which to me was very surprising because this is typically thought of as a lysosomal uh, enzyme. But when animals exercise, and humans too, it's released, its levels increase in the blood, and that's thought to be coming from muscle cells. And then when you administer these to animals or even in other types of preparations like cell culture, it can promote neuroplasticity. They can. IGF-1, definitely this insulin-like growth factor 1 promotes synaptogenesis and neurogenesis. And there is evidence it can get across the blood-brain barrier, which is also another consideration. Do these even get across the blood-brain barrier? But the studies are published suggest they do. And this is, I think, yeah, this is a ketone, beta hydroxybutyrate. Uh, and this ketone, and I'll talk about that in a little bit in this presentation. It's been mostly studied from the standpoint of fasting, where during fasting, at some point, usually in humans, 10 to 12 hours, your liver glycogen stores are depleted. That's the source of glucose, which is generally used first after you eat. And then when the liver glycogen stores are depleted, fatty acids are released from the fat cells and are transported into liver cells where they're converted to two ketones that can be used as energy substrates to generate ATP. One is beta hydroxybutyrate, the other is acetoacetate. I'll talk about that in a minute. And then the fat cells can also release certain things. And with obesity, for example, your abdominal fat cells tend to release pro-inflammatory cytokines, but with exercise, this anti-inflammatory cytokine interleukin 10 is released. In addition to adiponectin, which is shown in animal and cell culture models to promote neural resilience. Okay, so there's a number of really neat ways neuroscientists have developed to study neurogenesis. Early on, it was just injecting animals with, for example, radio-labeled thymidine, DNA-based precursor, DNA base, or um, bromodeoxyuridine, which will be incorporated into DNA. And then you can detect that with an antibody. So those essentially tell you how does the cell divided and replicated its DNA. But then more recently, things like using modified rabies viruses to 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 label any newly generated neuron that has been incorporated into a neural network. And there's been a lot of nice studies looking at the time course of generation of newly generated neuron from a stem cell, and then their differentiation and their incorporation into the existing circuitry in terms of what are the synaptic connections with other neurons intrinsic or extrinsic to the hippocampus and so on. So there's quite a bit known about that. I should also say that some newly generated neurons don't survive. So kind of like during normal brain development, at least half of the newly generated neurons die. A lot of that death is thought to be due to kind of a competition for a limited supply of target-derived neurotrophic factor. So for your neuroscience people that are in the graduate program, you may be getting a little bored with this because you know you know a lot of the basic things, but I think it's interesting to consider this in terms of the exercise, intermittent fasting, intellectual challenges. Uh, this study, I don't want to run way over time because I have a lot of slides. Essentially, what this study did is they showed that running wheel exercises enhances spatial learning and memory ability using a touchscreen system and looking at essentially what they do is initially you can, and this takes a long time, you can train a mouse to touch, say, a big icon, a big square on a touchscreen for a food reward. And then you can teach it to touch a smaller icon. And then you can teach it to discriminate between two icons that are separated by a large distance on the screen. Okay, so for example, it gets a food reward if it touches its nose to this one, but not this one. Then you move these icons closer, and they make more errors if you move these closer, which kind of makes sense. And the bottom line is the runner animals are better at this spatial pattern separation task. This is just another look at exerkines. That was in the other slide. Sirtuins have also been studied from the standpoint of caloric restriction, exercise, and aging and cell stress. And there's early work in yeast uh, discovering SIR2, which the mammalian homolog is sirtuin-1 or SIRT1. And then since then, many other sirtuins, these are the mitochondrial sirtuins. I think I mentioned SIRT3, which is a deacetylase in the mitochondria that seems very important for their function for healthy metabolism, control of free radicals, protecting mitochondria and cells against stress. I think I showed this slide before. These are a few of among many more proteins that have been shown to be deacetylated by SIRT3. And in this study, this study asked a couple of questions. One, will running wheel exercise affect the expression of SIRT3 in the brain of mice? And this looked at mainly in the cortex and hippocampus. And then the second question was, if SIRT3 levels are increased, which they are, then is there a role for that upregulation of SIRT3 and any beneficial effects of exercise on the brain. And so in the study, they showed that using antibodies that only recognize acetylated proteins, they showed that in SIRT3 knockout mice, there's hyperacetylation of many proteins, both in the cytosol and the mitochondrial fraction. Sorry, yeah, the whole cell and the mitochondria in particular. There's a few here. Turns out there are, well, it can be said for sure. There are some proteins that are actually released from mitochondria. And of course, you also get mitophagy going on, in which bits of mitochondria are moved to the lysosomes. Okay, and then that's this quantification. Then in cultured neurons, if they lack SIRT3, so these are embryonic mouse cortical neurons exposed to increasing concentrations of glutamate and to canic acid or NMDA, and looking at excitotoxicity, cell death. And bottom line is neurons are more easily killed via excitotoxic mechanism when they lack SIRT3. On the other hand, if one uses a viral vector to essentially overexpress SIRT3 in the neurons, then they're more resistant to excitotoxicity. In vivo, SIRT3 knockout mice, their hippocampal neurons are more vulnerable to epileptic seizure-induced damage. So this is injecting canic acid, which is it's an excitatory amino acid. The reason it's so effective in causing seizures and killing neurons is that it activates the glutamate receptors, but canic acid is not removed from outside the cell via the normal glutamate transporters that remove glutamate. So whereas in vivo you can inject a high amount of glutamate itself in the brain and not see too much happening, it's very difficult to kill any neurons because when you put that glutamate in, it's taken up by, for example, astrocytes and removed, whereas canic acid isn't. Um, right. In culture, if you activate glutamate receptors at a low level, well, even below that, that causes, uh, well, it's shown here, 1 micromolar. So this is a subtoxic level, uh, probably closer to physiological levels in this condition that increases SIRT3 levels. If you get very high levels, neurons are dying. Um, okay, so glutamate itself, which causes calcium influx, is upregulating SIRT3. And not clear which transcription factors involved. For example, it could be Krebs or others. And then in vivo, the running wheel exercise increases SIRT3 in the cortex and hippocampus. And so to get at the question of in vivo of this, peripheral versus central effects of the exercise. So in this experiment, a NMDA receptor antagonist, MK-01, was given. So idea is you block glutamate receptors in animals, and these are MK-01 at the right dose will cause kind of a partial reduction in activation of NMDA receptors. And the bottom line is that when you block NMDA receptors, the ability of exercise to increase SIRT3 expression is abolished. Okay. Now, one thing that was shown a long time ago, in fact, it was a system, at least to my understanding, where mitochondrial biogenesis was really first discovered and the mechanisms elucidated. So physical exercise in muscle cells will, over time, over multiple periods of exercise, rest, increase the number of mitochondria in the muscle cells. And that makes sense. Uh, then in the future, they can generate more ATP to increase endurance or strength. And so the pathways have been identified, at least in part, and they involve this AMP activated kinase. So um, kind of the AMP to ATP ratio is an indicator of kind of the bioenergetic state. Um, and so typically when ATP levels start to drop, AMP levels should uh, should increase, and then this kinase is activated. Also, the free radicals generated in the muscle cells in response to exercise have been shown to have a signaling function that can activate this transcriptional master regulator mitochondrial biogenesis, PGC1 alpha. Um, and there's a huge literature on this, many review articles just written on all this. So then the question becomes, does this occur in neurons in the brain in response to exercise or just exercise, either physical exercise or exercising the muscles, otherwise, for example, by environmental enrichment or intellectual or social enrichment? And the answer is maybe. Um, so as neurons grow, number of mitochondria increase and their ATP generating capacity increases. And by using a viral vector to knock down PGC1 alpha using RNA interference, it was provided evidence that if you do that in cultured hippocampal neurons, it inhibits synapse formation between the neurons. Okay, and then if you knock down PGC1 alpha, it also blocks the ability of BDNF to induce synapse formation. I'm not going over the details on this. The take-home message is what you need to get from this. And then in vivo, using the same RNA interference viral AV technology, PGC1 alpha was knocked down in hippocampus, and then dendritic spines were quantified on this is dentate gyrus. So there's GFP in the AV2, so you know which neurons are infected and then which, and then this was shown by immunostaining. It works. They have reduced PGC1 alpha, and in those neurons, there are significantly fewer dendritic spines, although it didn't affect the length or the branching. Okay, and then this is just looking at some potential ways that exercise, which I've talked about, intermittent fasting, which I'll talk about next, and cognitive stimulation um, may promote mitochondrial biogenesis. Some of the same overlapping mechanisms as with muscle cells, although the BDNF and Krebs, there is evidence for that pathway as well. All right, so it's been known for 70 years, actually, or there's been evidence for 70 years that if you reduce food intake in rats or mice, they live longer, and up to a point when they starve and die from starvation. Now, rats or mice can only go 3 days, or mice anyway, 3 days with zero food. They go any longer, they die. So they're very, with contrast with humans, which can go a month, 2 months. If there's been case studies in individuals that are severely obese, going like 6 months with no calorie intake. But anyway, um, when you reduce the calorie intake to a certain level, it's kind of bordering line on starvation. Uh, when that started, when the animals are adults, young adults, they can live up to twice as long, depending on the strain. And in this study, they showed with this kind of severe calorie restriction, the size of every organ that they looked at, heart, liver, kidneys, I don't think this, spleen, skeletal muscle decreased. What didn't decrease was the brain. No change in size of the brain, even though there's dramatic reductions in these other organ size. And the other organ that didn't decrease in size was the testes in males. And I have a theory for that. It has to do from evolutionary perspective that when animals are in an environment where there's no food around, um, uh, they don't have ovaries, but in the females, the females quit cycling. They no longer have estrous cycles, so they're no longer fertile with this severe calorie restriction. The males remain fertile. So if you're a female, you don't want to get pregnant because you don't have any energy resources to promote the development of the embryos. If you're a male, maybe you want to try to inseminate as many females as possible before you starve to death. So that's just a, hypothetical explanation for that. And in this study, that was done by a colleague of mine a long time ago at the NIH, Don Ingram and his group, they found if they subject rats to every other day food deprivation, so one day the animals have free access to the food for 24 hours, the next day you take the food away for 24 hours. And when that started when they're young, both the average and maximum lifespan are increasing dramatically. And these are the body weights here. So when the animals are younger with this every other day food deprivation, they do gain weight, but then it kind of plateaus throughout the rest of their adult life. The ad libitum fed animals that have food every day, they continue to gain weight, and a lot of this weight is fat, which is just obvious when you look at the animals in the cages. Um, so with in humans, our typical eating pattern is breakfast, lunch, dinner, maybe an evening snack every day. Uh, if one doesn't get any exercise at all and you eat this pattern, every time you eat, the liver glycogen stores are replenished. And so you never get the metabolic switch to using fats and then the ketones derived from the fats. This is like, and these are just uh, kind of examples based on a lot of data. That's someone that fasted completely for one day and then ate 3 meals the next day. And this is an individual who ate all their food within a 6 hour time window, so essentially skipping breakfast. And their metabolic switch occurs in the morning hours. If they were to exercise here, there'd be a bigger increase in ketones. Um, and same here, if this individual exercised here. If you exercise here, like if you get, whatever, get up in the morning, go on a run, then you might have the metabolic, you'll likely have the metabolic switch occurring during that run. Interestingly,
Excuse me, I have a question. So,
yeah,
Are these patterns the same for males and females, like in humans?
Uh, Generally, yes, they're the same. You
Okay,
mean
so
in terms of how they affect glucose and ketone levels?
yes.
Yes.
Okay, thank you.
Yeah. Interesting that the way pretty much all of the early calorie restriction studies were done was you say take 20 animals, they're individually housed, one animal per cage, and you measure the amount of food they eat, the weight of the food each animal eats each day, and you do that for a few weeks. Then you divide them into two groups, and one group you give, say, only you give them 30% or 40% less food than they would eat ad libitum. So that's the calorie restricted group. So that's in this study the 40% CR group. Okay, and then the control group is they just eat uh, ad libitum. And interestingly, when you do this, when you give an animal 40% less food than it would normally eat, it eats it all within like a 6 hour time window. And so it's actually fasting for up to 18 to 20 hours every day. And so that has raised the question of what's the relative contribution of calorie restriction per se to this fasting component and metabolic switching? And we don't know the answer. There's some evidence that benefits of intermittent fasting uh, can go beyond what can be accounted for by any overall reduction in calorie intake. So, for example, one can not have any change in weekly calorie intake and see some improvement in glucose regulation and other indicators. This just shows that in these animals, the ketones go up pretty quickly after the onset of fasting, within 4 to 6 hours of fasting. So they have the point is the animals have this metabolic switch much earlier than humans. Okay, so when I retired from NIH, I finally had time to write a book on the science of this. I didn't have time when I had a big lab and had to focus on the postdocs and graduate students and stuff. But there's a huge literature now. Most of it has come about during the last 15 years or so, because NIH put a lot of money into animal, but also many human studies of intermittent fasting. If you go on clinicaltrials.gov, there's over 200 ongoing trials in humans with various disorders, cancers. Some are oriented towards brain disorders. And so if you're interested in that, you can just go on clinicaltrials.gov and plug in intermittent fasting. Okay, so let's just quickly look at this metabolic switching. So glycogen depletion from the liver, so that's the glucose store, that triggers lipolysis, so the breakdown of triglycerides, diglycerides into free fatty acids, which are released from the fat cells into the blood. They're transported into liver cells and converted through this pathway. And you'll see this pathway, you get acetyl-CoA, which you'll know about from basic mitochondrial cell energy metabolism. So these two ketones, BHB and AA, there are transporters for them. So for example, in neurons, they have MCT2, monocarboxylic acid transporter 2, that transport the ketones in, acetoacetate or BHB, and then they're metabolized back to acetyl-CoA and goes into the TCA cycle to generate ATP. Studies have shown that this ketone in particular has signaling functions and acts like a hormone. So there are certain transcription factors that are activated. I think I have a slide describing some of the ways this may work. Uh, there's even evidence that this ketone can modify certain amino acids on proteins. Um, BDNF is upregulated both in cultured neurons and in vivo uh, by this ketone. And there's some evidence then for this mechanism playing a role in mitochondrial biogenesis in neurons. Right. So these are two studies, one in cell culture, other in vivo from a different lab, providing evidence that this ketone, beta hydroxybutyrate, BHB, upregulates BDNF at the transcriptional level. This slide was from actually, this is published, but uh, this just shows some of the pathways that may mediate effects of this ketone on gene expression. These are data from some early studies showing that every other day food deprivation uh, uh, done for several months can protect neurons against excitotoxic, uh, metabolic, and other toxic insults. So this is looking at canic acid model. This is looking at a stroke model, middle cerebral artery occlusion. This is looking in, well, there's a previous study in mice, and then this study in rhesus monkeys, providing evidence that um, actually this one was daily caloric restriction in the monkeys for 6 months, protected the dopaminergic neurons and lessened motor dysfunction in this MPTP model. And then this Huntington's disease model was Huntington mutant mice subjected long-term to every other day food deprivation. It's actually done by Wenjian Duan, she's a professor at Hopkins. It was a long time ago. Uh, and then also in this triple transgenic mouse model of Alzheimer's disease, which I think I mentioned before, long-term, in this case, 1 year of either daily time restricted eating or every other day food deprivation ameliorated the memory deficits in these mice. Now, one thing that's known about brain aging and cognitive impairment and Alzheimer's disease is there's reduced glucose utilization. So uh, uh, clinicians and scientists have taken radio-labeled uh, glucose, which is usually 2-deoxyglucose or no, yeah, 2-deoxyglucose, I guess. And so you inject it in a blood vessel of a human and then do a PET scan to detect the radioactivity. So in the normal brain, of course, red is a lot of uptake of radio-labeled glucose. Blue is very low or no uptake. These are the ventricles here, so they shouldn't have any uptake. Uh, in Alzheimer's disease, there's a dramatic reduction in glucose utilization using this method. This has also been shown in some animal models of Alzheimer's disease. Also, older individuals with mild cognitive impairment have reduced glucose uptake. And uh, oh, well, this this is a study showing that in cultured neurons, if you expose them to amyloid beta peptide at levels that cause a lot of stress and eventual death of the neurons, it impairs glucose uptake. All right. In this study, a group up in Vancouver, they did PET imaging using the radio-labeled glucose, 2-deoxyglucose. And then they also, in these individuals, they had a tracer for acetoacetate. So this cerebral metabolic rate acetoacetate here. And then they put them on a ketogenic diet, so no carbohydrates for several weeks. And so what you can see, there's a big switch in the individuals who are eating carbs, they're assumedly using the glucose, the neurons, but there's this big switch uh, when when glucose levels are depleted because presumably their liver glucose is depleted. And conversely, on the ketogenic diet, there's less utilization of glucose. So the point is, brain cells, and these are mainly nerve cells probably, because they're consuming the most energy, uh, can switch back and forth and you taking up these energy substrates. And there's a nice perfect correlation between blood ketone levels and the cerebral metabolic rate uh, for ketones. Uh, Turns out if you give in this mouse model of Alzheimer's, you supplement their diet long term with this ketone, beta hydroxybutyrate, it has significant benefits in lessening their cognitive impairment. And there's also efforts to kind of boost cellular neural energy metabolism using other approaches, which I'm not going to talk about, but one is this supplement with NAD substrates. This is a energy, very important for energy metabolism. It's also important for function of several of the sirtuin deacetylases. Uh, this just from the same study here, a test of anxiety. This is this open field test for anxiety, and the bottom line is the ketone reduced anxiety. You may know that ketogenic diets are used to treat epilepsy still in patients who don't respond to the normal drug treatments. And these ketones can suppress or at least prevent hyperexcitability. And that's why ketogenic diets are effective and may also reduce anxiety, perhaps through a similar mechanism. Okay, let's see. Yeah. I'm going to skip over this. Reduces anxiety. Yeah, so I mentioned intermittent fasting increases SIRT3 levels. Um, and this ketone that's elevated in response to fasting seems to suppress neural excitability. So in this study, uh, what was done is uh, patch clamp recordings from CA1 neurons, hippocampal CA1 neurons, recording essentially GABA currents, inhibitory postsynaptic currents. So recording from an excitatory glutamatergic neuron, the activation of GABA currents, which are the chloride channels. So you get these downward deflections, which indicating opening of the chloride channels. So let me see, I got to remember what this, right. Yes. So the bottom line is that intermittent fasting will increase the GABAergic tone, if you will, perhaps explaining, at least in part, beneficial effects. I'm going to skip that. What about humans? Uh, this study came out recently. It was done by Dimitrios Kapagiannis, a neurologist at the NIA, actually, with kind of my cheerleading and a little bit of quite a bit of help in designing the study, but otherwise I wasn't involved at all. Um, right. So what was done here is individuals who are at risk for cognitive impairment because of their age and metabolic status, they were 55 to 70 years old with obesity and insulin resistance, but not frank diabetes. And they were randomly assigned to intermittent fasting 2 days a week, eating only 480 calories, which is hardly anything, and for 2 consecutive days. So on these days, the ketones were up. This, the control group was, they did the USDA healthy living diet, which is uh, calorie reduction. Uh, the idea was to match the weekly calorie intake of these two groups by having these individuals eat 3 meals a day, spaced out, but each meal had less calories than they normally eat. And so when did the math, was predicted they'd have pretty similar weekly calorie intake, what seems to be the case. Then there's a lot of things done on these, a lot of basic measurements of body fat, glucose regulation, a lot of blood work, and then cognitive testing focused on various domains of cognition. Um, and then some analysis of the brain, structural MRI. There's this group, uh, another lab developed this brain age index, which is kind of what I talked about in the previous lecture, that, um, are there biomarkers of brain aging? And so this is kind of MRI-based biomarkers of brain aging. And then I think I talked about, maybe I can't remember, extracellular vesicles. Anyway, turns out in the blood there are these small, like say 100 nanometer diameter vesicles that they come from a variety of cells, but there's subpopulation from neurons that can be isolated using antibodies and probed. So there was some analysis there. And so these are the anthropomorphic measurements. These are the ketones, uh, which are up more in the intermittent fasting group. Um, there's reductions in some, or improvements, if you will, in some cardiovascular risk indicators, total cholesterol, LDL, and improvements in glucose regulation. So this is an indicator of insulin resistance. So lower is better here. And there's a bigger, statistically significant bigger improvement in the glucose regulation with the intermittent fasting. And then in tests of this basic memory, can you remember how many words on a list can you remember on a list that you looked at a half hour ago? Um, verbal fluency, uh, task shifting, how well can you shift your attention from one task to another? And the interesting thing here was there were significant improvements with the intermittent fasting group, but not the control group, which was the healthy living diet. Okay, so I'm almost done. Now, in addition to yeah, so I put up on Canvas this morning the article that has the following two figures on it. And essentially, they're saying this one says that there's this general idea that challenging the brain metabolically, cognitively, and free radical production with increased activity, uh, intermittently can optimize health. So during the challenge phase, for example, fasting and exercise, evidence is emerging that cells activate pathways that promote cellular stress resistance and kind of conserve resources. So, for example, there's downregulation of mTOR and overall protein synthesis, but upregulation of uh, genes encoding things that promote autophagy, DNA repair, antioxidant enzymes, for example. And then during the recovery phase, eating, resting, sleeping, cells go into a growth and plasticity mode where mTOR goes up, protein synthesis goes up, mitochondrial biogenesis goes up. Uh, so the idea here is these conditions prepare the cells for future stress and set in motion changes that occur during the recovery period that enable or mediate plasticity, whether it's synaptic plasticity, neurogenesis, and you have to have switching back and forth between these challenge recovery cycles over periods of weeks, months, years. So this is the working model, and here's some of the specific pathways here, which I've talked about and you know about. And yeah, yeah. So anyway, here's for example, contrasting uh, moderation in energy intake, perhaps intermittent fasting, exercise, cognitive stimulation with metabolic complacency. So the idea is that if you're overloading the system with energy continuously and amino acids and so on, and you're not challenging your brains with intellectual challenges, for example, they pathways that evolved to promote stress resistance, resilience are kind of downregulated. Uh, okay, well, got 10 minutes. Okay. So this is a kind of the general idea. There's this concept of hormesis, which is just another, it's in a way, a synonym for adaptive stress responses. So the idea is that cells, organ systems, organisms respond to various types of physiological stress. It could be heat stress, cold stress, food deprivation, uh, physical stress, um, chemical stress, uh, within a certain range of exposure doses and durations, the systems respond adaptively. And and then if those challenges are repeated, promote health. So there's this called hormetic zone that can enhance function, fitness, disease resistance. To go beyond that can cause problems. Now, it, it's hard since we evolved to adapt to things like exercise and food deprivation. It's actually hard to go beyond this unless you really, really want to. Right. So, for example, exercise, you run to exhaustion, right? And typically individuals aren't going to kill themselves when they don't have to. Uh, and then possibly by combining different physiological challenges intermittently with spacing. There hasn't been much work on this done, for example, combining exercise with intermittent fasting, exercise with intellectual challenges, exercise with sauna, you know, exposing yourselves to heat intermittently. It seems to have some good effects. I did want to point out that so we're omnivores, and a lot of animals are herbivores, and these plants produce chemicals that prevent, for example, a fruit that's not completely ripe from being eaten. So, for example, in tomatoes or I don't know what kind of fruit this is, it doesn't matter. The unripe fruit is green, very bitter tasting, uh, not really too palatable. Then, as the fruit ripens, and also importantly, from the standpoint of the plant, the seeds in that fruit are not yet mature. Then, as the fruit ripens, uh, more sugary substances start to accumulate in it, and the amount of the chemicals in the skin of the fruit that have a very bitter, kind of noxious taste decline. So then these are more palatable. Then the monkey or we or a bird or whatever eats the ripe fruit, which has seeds, and then, some of them may be dropped on the ground there, or they may go through the GI system. Many seeds can get through the entire GI system and still be able to germinate. So this coevolution is kind of interesting, and the monkeys may be actually helping the plant disseminate itself, and the yeah, so it's pretty interesting. On the other hand, even less ripe fruit has some energy and nutrient value, so it it's we evolve ways to tolerate exposure to these chemicals. Uh, yeah, so for example, some of these chemicals, caffeine, you know about capsaicin, which is in actually hot peppers, epicatechins in green tea and so on, sulforaphane in broccoli sprouts. There's a lot of stuff in the lay media about this, for example. All of these have very bitter taste, and if you, for example, put tea leaves or coffee beans or hot peppers or broccoli or broccoli sprouts on your kitchen counter in the summer, you will not find ants uh, eating them. However, if you were to put a ripe fruit or uh, well, some other things that don't have these chemicals or high concentrations in them, the ants will eat them. So that's kind of a I don't know. This is kind of an observational thing. Uh, and it turns out that any chemical which seems to have beneficial health effects from plants does have a bitter taste. And there are at least four ways we evolved to prevent from overdosing on any of these. One is very bitter taste or hot taste. Another is are vomiting. The third is there's enzymes in our liver that rapidly degrade these since we co-evolved with them. And that contrasts to man-made pesticides, for example, which we didn't evolve with, and the enzymes in the liver don't get rid of them, so they stay in our system long time. So when we ingest these, we tend to metabolize them and pee them out pretty quickly. And then the fourth mechanism is that these chemicals can induce adaptive stress responses in cells. So, for example, sulforaphane activating a well-known pathway called NRF2 transcription factor, which upregulates antioxidant enzymes, for example. Okay. I wanted to say one last thing. There's some really interesting work that we'll see what happens in the clinic. That there was an initial safety trial, but uh, group out at Stanford, Tony Wiscore, but others too, Rondo, uh, have provided evidence that if you transfuse old mice with blood from young animals, it rejuvenates various organ systems, uh, including the brain. And I'm not going to go into any detail on this. It's pretty fascinating. Uh, Maybe too good to be true, but I don't know. We'll see what comes out of it. So there are actually, there's people, usually rich people in the lay public there's one individual that I am aware of that he is having his kids donate blood and storing it. You can store it obviously for extended time periods for his own transfusion so he can be rejuvenated. But anyway, this is just early days on this, and the extent to which this is something that would ever, number one, is it really effective, and two, what's the practicality, and ultimately we need to know what are the mechanisms. They are providing some evidence for mechanisms. Okay, so I'll stop there, and thanks for your attention.
Thank you.